Welcome, everybody, and thank you very much for joining us today for today's webinar hosted by the Society for Neuroscientists of Africa. My name is Arif Hamid, and I'm a research fellow at Brown University in the US. It's really my pleasure to introduce Dr. Udunna Anazodo, who's a PET MRI scientist and principal investigator and assistant professor in the Department of Biophysics and Clinical Neurological Sciences at Lawson Health Research Institute at Western University in London, Ontario, in Canada. Dr. Anazodo received her bachelor's degree in psychology and biology from York University in Toronto, and also medical radiation sciences from the University of Toronto. Dr. Anazodo directs the Mind Lab, and her research focuses on developing non-invasive PET and MRI techniques for clinical applications in brain imaging, with an emphasis on combining the strengths of PET and MRI to enable novel and in vivo measurements of brain function. I'm really excited to hear about her work today, and I just want to remind everybody joining us live to put your questions in the chat window so that we can ask them as part of the Q&A discussion at the very end of the talk. So Udunna, without further ado, the floor is yours.
Thank you so much for that wonderful introduction. I'm really happy to be here, and I'm going to try to keep this to that 45 minutes so I could get to all the questions because I love to interact with the audience. Can you guys hear me? Yes? Okay.
Yes.
All right, so I want to thank the organizers for the invitation to give this SONA webinar series, and I very much appreciate the opportunity. So today I would like to briefly discuss the emerging role of Atherospin labeling MRI in neuroscience, and to show that MRI can be a reliable in vivo imaging technique for probing changes in brain health. So the brain is my favorite organ in the body, and it's pretty much because the brain is very intriguing and a very unique organ. It weighs only 2% of the total body mass, but it consumes quite a lot of the total oxygen and blood glucose available in the body. It uses roughly about, I would say, 15% of the total cardiac output to meet this demand. So we can see that the brain is highly metabolic. It's always on. So even when you're asleep, the brain is still awake and working to regulate key body functions, which means that the brain needs a constant, steady supply of blood flow. Now, much of the blood flow supply, so about 60 to 80%, goes to neuronal signal processing and regulation of neurotransmission. Now, this is well demonstrated in this seminal work by Drs. Peter Fox, Marcus Reichel, and Mark Mitten, where they showed that viewing a visual stimuli like this checkerboard evokes a focal neuronal activation in the visual cortex, producing a significant focal increase in blood flow and glucose metabolism or glucose consumption in the visual cortex, so it's very focal, without any accompanying increase in oxygen use or metabolism. So the energy or the supply, the substrate that basically drove this activation came from blood flow supply, local blood flow supply, meaning that the ability of this subject's brain to process this visual stimulation was a result of a local increase in blood flow supplying the required oxygen to meet the local energy demand and obviously process this visual stimuli. Now, this is true for almost all brain, actually not almost, for all brain activities. So for example, a motor function shown here, and this is a finger tapping experiment showing an increased focal blood flow in the primary motor cortex in response to that finger tapping activity. And this is also the case in response to cognitive demanding tasks or emotional processing. So this means that the brain has to maintain a tight coupling of blood flow to neuronal activity. So an increase in neuronal activity will lead to an immediate increase, a local increase in blood flow and glucose consumption. Now, the brain has developed a complex regulatory mechanism to ensure that this tight temporal and spatial, so regional, link between neuronal activation and blood flow is maintained at all times and tightly regulated. Now, it does this through an autoregulatory mechanism that ensures that the brain has a constant and steady supply of blood to redistribute to sites of local demand. And to maintain this constant, steady supply of blood or autoregulation, the brain has developed a neurovascular unit to regulate vascular tone. So it vasodilates in response to an increase in neuronal activity. And I mean, on a microscopic level, the neurovascular unit is essentially made up of vascular smooth muscle, the neuron, and astrocyte glial cells. So for those interested in learning more about neurovascular unit and its function in brain health, I really love the neurovascular unit, but for those of you that are interested and want to learn more about it, there's a very nice overview by Professor Ida Kola that was published about 2 years ago in Neuron. I recommend that you read that. I mean, I would love to go into detail with this, but we're gonna move on for the sake of time. So throughout the human life span, cerebral blood flow, or CBF, is tightly coupled with cerebral metabolic rate of glucose consumption, or what we call CMR glucose. And this neurovascular coupling makes CBF an ideal index of local neuronal activity. In several brain disorders, for example, in Alzheimer's disease shown here, we see matched deficits. So we see hypermetabolism accompanying hyperperfusion. So we see matched deficits in CBF and matched deficits in glucose metabolism. And we nicely demonstrated this here. So during my postdoc work at Lawson, we followed a cohort of frontotemporal dementia patients where we demonstrated using simultaneous measurement of blood flow and glucose consumption that the deficits that we see in CBF, so in blood flow, is matched to what we see in neuronal activation here demonstrated with glucose metabolism. So there is this co-localization or matched pattern of hyperperfusion and hypermetabolism, again demonstrating this neurovascular coupling. And if local supply of blood flow, for example, for any reason is down, then we can expect impairment in local neuronal activity. So this sort of highlights the significance of CBF, so cerebral blood flow, as an index of local neuronal activity and overall brain health. Now we're beginning to understand that the relationship of CBF to glucose metabolic rate or the rate of glucose consumption, CMR glucose, may not always be one-to-one or matched. So neuronal metabolic demand may sometimes be unmatched by blood flow support. And in some cases, there may be changes in glucose metabolic rates, or CMR glucose, such that there could be an increase or decrease in glucose consumption that may not always occur with measurable change in blood flow. And we recently demonstrated this uncoupling of CBF in a meta-analysis study in schizophrenia patients where we found matched. So here in blue, we're showing both an increase in glucose metabolic rate and an increase in CBF. And in the red, we're showing areas of the brain that showed concomitant decrease in glucose metabolic rate and decrease in CBF. But there are also key areas of the brain that are very implicated in the pathogenesis of schizophrenia, like in the prefrontal cortex, where we didn't see this concomitant or matched or coupling of this neurovascular coupling, so coupling of blood flow with glucose metabolism. And some emerging, I guess, possible reasons or rationale for that could be because of vascular dysregulation that's already happening, where the brain is no longer able to provide the matched blood support to sort of meet the neuronal demand. And indeed, this vascular dysregulation is implicated in Alzheimer's disease and has been shown to be one of the earliest indicators or biomarkers of Alzheimer's disease. So this is a study by Yasser Mendina from the MNI. And this is a very nice study demonstrating how changes in blood flow could precede or can precede in case of Alzheimer's disease, changes in memory. So that's the behavioral changes that would tend to bring people to the Alzheimer's clinic, which is shown here in green, or even changes in metabolism, glucose metabolism shown here in blue, or even changes in brain atrophy, which is shown here in atrophy is structure, light blue. And so we see that blood flow changes are happening much sooner. So they do and can precede changes in neurodegeneration, or they can precede neurodegeneration and start way long before overt behavioral changes like memory actually manifest. And we've sort of done something similar, not to the extent of what Yasser did. So this is a study I did for my PhD where I looked at the effect of cardiovascular disease on the brain. So we're looking at the effect of cardiovascular disease on brain health. And we wanted to look at blood flow changes because, again, we think that blood flow changes are an indication or a good index of brain health. And so what we found was that coronary artery disease patients who had no overt changes in cognitive function, so overall, their cognitive function is like, similar to their age-matched controls, showed concomitant hyperperfusion and brain atrophy. So we found areas of the brain that had brain atrophy, so loss in gray matter volume or neurodegeneration, also had hyperperfusion, but not in all areas. In some areas of the brain, so in some areas here, we see that, well, we found that there were changes happening in blood flow, but not matched with gray matter volume loss. So the brain structure looked normal. Again, suggesting that changes in blood flow and vasculature could potentially precede changes in neurodegeneration. And again, long before overt behavioral changes manifest. So we can see that, like I said, CBF and trying to measure CBF and the vasculature is very, very important in determining or trying to understand or even assess brain health. So maintaining a healthy supply of blood flow is important in maintaining brain health. So then how do we measure or monitor changes in brain health in live humans? Now, there are several techniques out there used in measuring changes in cerebral blood flow. And some of them are based on administering an exogenous, so a contrast agent or a tracer. And some like Atherospin labeling ASL are completely non-invasive, so it doesn't require the administration of an exogenous or contrast agent. And ideally, if you want to sort of measure brain health and study it long-term and monitor it long-term, so look at potential early changes, so as an early biomarker, look for treatments, so being able to assess efficacy in treatment, for example, or even just looking at prognostics. Ideally, you want a technique that is robust, so it's highly reproducible. You can do it multiple times in the same person and sort of get ability to get similar results. In a healthy person, if you were to scan the brain multiple times, you want to get the same blood flow measurements. So you want a technique that's very robust. And you also want a technique that is sensitive enough to detect early subtle changes. So like I mentioned earlier, for example, in the coronary artery disease patient study that we did, where we saw changes in blood flow without changes in brain atrophy or gray matter volume, or even any sign of memory loss, same as the MNI, large MNI study in Alzheimer's disease, where they're seeing early changes in blood flow, but not always changes in neurodegeneration or cognitive impairment. And so you want a technique that is sensitive enough to detect subtle changes and detect it early enough. You also want a technique that is non-invasive, so it can be safely used to monitor and track changes in sick patients. So you want it to be well tolerated. And that almost eliminates all contrast agents because they're quite invasive. And you want a technique that will provide regional measurements. So you don't want to go for global total blood flow like what transcranial Doppler ultrasound tend to do. It doesn't give you that regional measurement. So you want to be able to see what's happening on a local or spatial or regional level in the brain. Ideally, you also want an imaging technique that is multiparametric, so that can give a little bit more information than blood flow measurements, because you also want to be able to assess what's going on in the vasculature. So what's going on in terms of vascular tone, how well the brain is able to regulate blood flow supply. So if it's able to vasodilate and vasoconstrict on demand. And if it's, if there is a breakdown in blood-brain barrier, for example, a breakdown in vasculature, you want to be able to also get that as an additional information. So it'd be ideal to have a technique that somehow allows you to do all of this together in one measurement. And lastly, you ideally want a tool that is easy to use, so it's readily accessible. And so there'll be no barrier due to cost or infrastructure that will limit access. So you want to be able to do this type of measurement. So you want to be able to get information on brain health across all populations, irregardless of where they are and what access they have. And so there's quite a lot of techniques out there for measuring blood flow, but not all of them meet all this, what I call ideals for being able to get a tool that can ideally give us brain health information in almost any population. So now the current gold standard for measuring blood flow is using radioactive water. And that's done with positron emission tomography or PET. Now, although PET is robust, sensitive, and provides regional measurements, because essentially you are injecting a radioactive tracer that is easily diffusible, it's water, in the brain. So it provides you a very sensitive measure. It's very robust. It gives you regional measurements, but it's radioactive water. So it's highly invasive and it's expensive. You need a cyclotron to be able to produce the tracer. And for O15 water, so for radioactive water, the half-life, it's 2 minutes. So, you need to get the tracer made, take it up to your scanner, inject it and scan. And in that time, your tracer is decaying. So which means that there's only a handful of sites in the world, like the one like ours here at Lawson, that has a cyclotron that's within a short distance from the scanner and can allow us to perform blood flow measurements with this gold standard technique. So although it's gold standard, it's very expensive and it's very highly invasive. So if you want to do long-term monitoring, for example, you want to follow patients over time, you want to look at treatment efficacy and stuff like that, you have to give multiple. You have to expose your patients or your study participants to radioactive water multiple times. So it's not really ideal. Now, on the other hand, Atherospin labeling is an MR technique. Oops, there goes my scanner. Sorry. So on the other hand, Atherospin labeling is an MRI technique that allows direct measurement of regional blood flow without the need of any exogenous agent or contrast. So you don't need to inject anything. You don't need a cyclotron. You need your patient or your participant and a scanner. Now, so if we were to compare ASL to the gold standard, because I think ASL is that near ideal tracer, it's robust. It gives us direct measurement of regional blood flow. And like I'll show in a couple slides, the next coming slides, is comparable to the gold standard PET. And it's also non-invasive and readily accessible. So like I said, you don't need an MRI scan. Sorry, you don't need a cyclotron. You don't need all the resources that comes to the cyclotron. You just need your MRI scanner. So compared to, just go here 1 second. Give me 1 second. Okay, so if we were to compare ASL to the other techniques, so we have DSE, which is dynamic susceptibility contrast imaging. It's an MRI technique that uses a contrast agent. We have PET, the gold standard with O15 water, which again, radioactive. And we have transcranial Doppler ultrasound, which is just a global index. It doesn't give us original measurements. So I'm just gonna X this one out. And if we were to look at the two MRI techniques, so we can also do DSC or dynamic or contrast enhanced imaging with CT as well. But if we're just to compare the two MRI techniques, ASL and DSC, we see that it does provide us a good temporal resolution that we want to be able to capture changes quickly and rapidly over time. It gives us regional measurement, but again, is invasive. You need to inject a contrast agent, and contrast agents are not readily available in all locales. So it's not readily available in a lot of places. So then if we were to, I'm going to X this out. Then if we were to compare ASL with PET, the gold standard, we see we have direct measurements with ASL and PET, because as I'll show very shortly, the ASL also uses blood water to generate its contrast to image blood flow. So it's, again, it gives you that direct measurement as radioactive water. It gives us regional measurements, like I mentioned, and bingo, it's non-invasive and it's highly accessible. And I'm going to go through this quickly, but we've done a lot of studies here demonstrating that ASL gives us very similar information to the gold standard O15 water and to what we consider to be the gold standard for brain health on neuronal index, which is measuring glucose metabolism with PET. Because we have an integrated PET and MR system. So here in Lawson, we're one of the few places, and the scanners are now becoming quite available, but we're one of the few places that have this integrated PET and MRI system where the detector is embedded within the scanner, the PET detector is embedded within the scanner. So we can do simultaneous imaging of PET and MR, and we can do this at the same time in the same subject. So because we have this scanner, we wanted to really validate ASL and demonstrate that it can provide similar information as the gold standard PET for blood flow, and also a good proxy index for measuring brain health, which is, comparing it to the gold standard for neuronal activation or being able to look at neuronal activity, which is using glucose metabolism measured by PET. And we've done these studies in frontotemporal dementia patients. And here we show that, we are able to get on an individual level, matched or similar patterns of deficit. So we have hyperperfusion that's matched to hypermetabolism. And, Tracy, the PhD student, Tracy Sassali, PhD student here at the Institute under my former supervisor, Dr. Keith Lawrence, she's done a series of studies where she's measured blood flow using ASL and the gold standard O15 water, radioactive water in healthy controls and also in FTD patients. And again, she's kind of showing similar patterns that we are able to detect on an individual level, and as well as on a group level, matched patterns of deficits, again, demonstrating that ASL can in fact give us similar information to the gold standard O15 water. I'm going to skip through this for time. So how do we perform ASL, and how does ASL allow us to measure blood flow directly without the need of an exogenous agent, so without having to administer a contrast agent? So with ASL, essentially we use arterial blood water. So arterial blood water is magnetically labeled, and we aim to apply a magnetic inversion in an area that's proximal to our imaging slice. So in the brain, our area of interest is our brain tissue, right? That's what we're interested in. So we apply a magnetic pulse in an area that's proximal to that. So we aim for vessels within and around the neck area. And so arterial water is magnetically labeled proximal to the imaging slice by a magnetic inversion. And then we wait for a time delay. So we wait to allow those magnetic spins to travel up naturally into the brain and exchange with brain tissue. And so we wait, we call this time delay a post-labeling delay. That's just essentially allows the label to travel up to our imaging slice. And then we acquire an image. And so this is an image of a tissue that's labeled with blood water. Now we do this again. So we acquire an image, but this time around, we don't actually apply any pulse or label. We just acquire a native image of the brain. And this image then becomes an image of the tissue with relaxed. So the brain obviously has its resident blood water. And so we basically acquired a second image that has a tissue plus the relaxed blood water. And essentially the difference between these two images, so the difference between the tissue relaxed and the tissue with the labeled blood is essentially our ASL signal. And then with a series of mathematical modeling, we convert that ASL signal into a blood flow image. And so going from, from here all the way to here essentially gives us a direct way of measuring blood flow without a contrast agent, because again, we're manipulating the arterial blood water that's resident within the person's body, in this case in the neck, and allowing that to exchange with tissue, and essentially trying to get a clear index or a clear index of brain tissue that's been perfused by blood flow moving into the brain. And again, by using mathematical modeling, we can convert that into blood flow. Now, there are several ways to label or magnetize arterial blood water. Each of them have its own inherent advantage and disadvantage. I'm not going to go through this because of time, but essentially, I just want to point this out to you in case you start reading up on ASL and you come across all this acronyms. So pulse arterial spin labeling or pseudo-continuous ASL or velocity selective ASL. These are all just different ways of being able to generate our magnetized blood water. And typically, the pseudocontinuous or the P-cancel is one of the ones that are very common and highly recommended because it has a relative higher signal-to-noise ratio. Now, there's also different ways of reading out that measured signal in tissue. Again, there are pros and cons of each of these readouts, but essentially, the idea is to use a readout that works well with your system or with your scanner. I guess in essence, the three-dimensional readouts are sort of preferred over the two-dimensional readouts because you get a whole brain coverage. And the two-dimensional readouts, you have to select which areas of the brain you want to target, but with the three-dimensional readout, you can sort of image the whole brain at once. And the three-dimensional readout is essentially what's becoming quite common now. Now, regardless of the signal label or readout approach, all these techniques provide a way to directly acquire regional blood flow information non-invasively. Essentially, when you're looking at a normal ASL blood flow image, you're looking for just homogeneous distribution of blood flow in the brain. So you're looking for a homogeneous blood flow pattern, and it's quite common. It's natural for you to see, if you compare a child to an adolescent to a teen, kids tend to have higher blood flow. That's just not natural compared to a teen, and a teen compared to an adult. But again, all you're looking for is this nice, normal, homogeneous pattern of blood flow. And like I said, regardless of the signal label or readout, they all tend to give us this homogeneous, nice pattern of blood flow. And ASL has demonstrated application in neurological disease or disorder management. I'm showing you sickle cell. I think it's relevant to us. And here's an example in healthy control in the sickle cell disease patients. And with ASL, we can start to get some information about the disease. And there's one of the telltale signs in sickle cell is this dural venous hyperintensity, this bright spot here within the dural venous sinus. That's a telltale sign for sickle cell disease. And this could be an indication, in this particular case, could be an indication of capillary shunting. So ASL does provide a way of using or measuring brain health, or in this case, blood flow in sickle cell disease. And there's a lot of other diseases where ASL has been applied to. I'm just picking and choosing a few because of time. Here's an example in stroke. In stroke, the current clinical standard is to do either CT perfusion, so use a CT contrast agent and acquire a blood flow image, or to do dynamic susceptibility contrast enhanced MR. So again, using a MR contrast agent to acquire a blood flow image. And here, this study, they're basically demonstrating that ASL provides similar comparable information to this, I guess, invasive or contrast enhanced agent techniques, and provides concordant information to all this series of clinical measures. And there's so many other cases, moyamoya disease, Alzheimer's disease. There's a lot of clinical cases where we're now seeing that ASL does have some application in the clinic. Now, if you recall from all the slides I showed earlier, were actually images acquired with ASL. So here again is that Alzheimer's disease study from Jin Chen's group at, I think at the time she was at Harvard, now she's at Baycrest in Toronto. But she basically showed, and this was quite early on, back in almost 10 years ago, that measuring blood flow with ASL and comparing that to glucose metabolism measured with PET gives you basically similar information. And I've discussed what we did here in London. Again, this was done with ASL imaging. I mentioned to you earlier on about looking at brain health, so measuring blood flow in coronary artery disease patients. This was done with an ASL MR technique. And the study looking at neurovascular uncoupling in schizophrenia disease, we looked at actually studies that either did ASL for blood flow measurement or PET glucose metabolism for looking at changes or measuring changes in glucose metabolic rate. And this study by Thierry Medina was actually done on thousands of brain from the Alzheimer's disease, so the ADNI, Alzheimer's Disease Initiative project, and this was an ASL study. So ASL has a lot of applications in both clinic and also in neuroscience. But if you remember when I was listing my ideal tracer, I talked about reproducibility or being robust. And we've done studies here in London. This is again study by Tracy and my former supervisor, Kit Lawrence, showing that ASL is truly robust and reproducible. So Tracy essentially looked at baseline and blood flow measurements during a task. Here I'm showing you baseline information, and she had a few healthy controls go in and out of the scanner. So we did a week apart and a month apart, and she looked at absolute blood flow changes, and essentially they look similar. The total gray matter blood flow measurements are, pretty much bang on similar. And even if you look at within session variability, the within session variabilities are quite low. I think the highest that we found was just under 10%, so shy of 10%. So this again demonstrates that ASL, at least when you're looking at baseline, just looking at blood flow on the baseline level, it is quite reproducible, so it's quite robust. This is, in the same study, in the same patients, Tracy essentially also looked at finger tapping, so looking at being able to detect changes in blood flow. And so she demonstrated again, if you were to measure these changes in blood flow within a session, so you have them come in and out of the scanner, you have them 1 week apart and 1 month apart, you pretty much get similar blood flow patterns, except for this person. Oh, I think it's me, I don't recall, I may have fallen asleep in the scanner. Because the finger tappings were for so long. But in essence, you do find that there are similar blood flow changes. And even when you try to correct for this by, say, using the relative measure, so by normalizing your blood flow measurements, say with the global blood flow index, you find that it does provide similar index or similar measurements, I would say. So it's quite robust. I'm going to skip this quickly, and this is an example of its robustness in a stroke. I think this is a glioma study, looking at recurrence of tumor. And essentially, this was ASL acquired on a 3 Tesla magnet MRI scanner. This is a contrast-enhanced MR T1 image. And this is the same patient rescanned on a 1.5T, so slightly stepped down in terms of signal strength, in terms of field strength, which obviously will sort of downgrade your spatial resolution. But again, we find that ASL gives us comparable information. So there's this intra-operative or intra-scanner reproducibility. And then this is during surgery and after surgery. And so this kind of demonstrates that ASL can be used for long-term monitoring and being able to provide some clinical information that could guide treatment planning. And so for the sake of time, again, I just want to make sure we have 45 minutes. When I was listing off those ideal list, I mentioned multi-parametric. So being able to extract multiple information from the same technique. So looking at blood flow changes, looking at vascular tone, so vascular reactivity, the ability of the vessels to contract and dilate on demand, and also being able to get information on vascular integrity or blood-brain barrier dysfunction. And now we're able to do some of these techniques with advanced ASL imaging. And so this is a study that came out a couple years ago and sort of took the MR world by storm. This is a study done by Yolanda Ohene, who is at UCL, where they demonstrated in a mice model that they can sort of use ASL technique to measure permeability. So to pretty much measure water exchange or blood-brain barrier dysfunction. And we're trying to optimize a technique, not quite similar to this, but similar to this, here in London with a group based in Germany and Amsterdam, where we're trying to optimize this to do imaging in humans, and also be able to pull out blood flow images, as well as looking at brain barrier dysfunction, so permeability, in a water exchange across the brain. And we're trying to make sure that this technique. So our ASL technique is very sensitive to even measure changes in dementia patients. And lastly, in terms of being able to provide this multi-parametric information, we've shown that ASL can be used to measure vascular tone. So looking at vascular activity, essentially change in vascular tone from, say, baseline to when an agent is given that will cause the blood to vasodilate. In this case, we used carbon dioxide, which essentially just causes global vasodilation. And we showed in cardiovascular disease patients that we're able to sort of detect changes across the brain on a regional level with this ASL technique that actually allows us to sort of start to see potential early signs of blood flow changes that are even changing, or vascular changes that are even changing long before blood flow changes are even visible or measurable. And so the question is, how far, like ASL right now is still a research tool. It's really not used in clinic. And the question is how far away, like what is sort of keeping ASL from entering into the clinical sphere? A lot of the application for ASL is in neuroscience, but it has a lot of promising applications in the clinic. And there's been a lot of studies that have demonstrated, a lot of these are comparable studies comparing ASL to other techniques, other blood flow imaging techniques that are used in clinic. So FDG PET, for example, which is used in Alzheimer's disease as a marker of brain health. And I think most of the studies are actually with FDG, so that's also O15 water, again, the gold standard. And there's a few studies that also looked at contrast enhanced imaging, so techniques that are currently available in clinic. And so this review sort of summarizes some of these comparable studies demonstrate that ASL provides comparable information to these other blood flow or even brain health imaging techniques. Here's again in the same review paper doing the same thing in ischemic stroke patients. Again, in clinic, we tend to use contrast enhanced imaging. And there's a few studies now that have shown that ASL provides comparable information. And again, in brain tumor imaging as well. Again, comparing ASL to current techniques that are available in clinic, demonstrating that it is providing comparable information. And so there's now been a push to sort of try to standardize. I mentioned the fact that there are different labeling schemes, different readout schemes. And so these differences sort of are one of the reasons that prevents ASL from sort of entering into the clinic, because you want to be able to standardize your measurements and be able to get, what's similar across different scanners or vendors or different techniques. So if a patient were to come to a particular location and you want to acquire an ASL image, you want to be able to have a standardized protocol approach for doing that. And so there's been a push towards standardizing ASL. This paper was published back in 2015, is the ASL white paper, essentially summarizing some of the parameters that if you want to acquire a robust, sensitive ASL technique for use in clinics, especially in older patients, or even in kids, there are specific optimized parameters that are suggested. And another reason why ASL is sort of struggling to make it into clinic and has been more or less a research tool is because of the complexity in terms of being able to generate this blood flow image. And there's issues with being able to do quality control, correct for motion, remove some outliers in your signal. Sometimes these outliers are due to head movement, and ASL is very sensitive to motion. And in patients like in Alzheimer's disease patients, you want to be able to measure blood flow and not have it confounded by other vascular information. So being able to do this white matter hyperintensity or white matter disease correction, for example. So these are very complicated analysis techniques, but they're now approaches that have now been able to combine all these techniques into a GUI push buttons, simplified approach. And one of these approaches is Explore ASL, which I was happy to collaborate with. And this is a tool that was developed by Henk-Jan Mustarts and Jan Petra. And essentially, this tool is one of those tools that are now being being put towards a easy to use multi-vendor. So it doesn't matter which ASL MRI scanner that you have, which ASL technique that you have. So it doesn't matter which labeling technique, or which readout technique, this tool is able to analyze your ASL image. The tool from the group in Oxford, Michael Chappell's group, they have this quantifies tool that basically allows you to do ASL analysis on a push button sort of easy to do workflow. So all these approaches are sort of, you know, bringing ASL more and more towards the clinic. And this is an application of this Explore ASL analysis tool. It's been used to look at HIV in children, to look at cognitive impairment in elderly adults, and also to look at sickle cell disease. So I'm just mindful of time. In terms of, again, trying to push ASL into clinic, there are so many recommendations out coming out right now, giving neuroradiologists a guide, a way to sort of start to read and interpret ASL images. Again, these are welcomed improvements or welcomed approaches that we now can sort of bring ASL more and more towards the clinic. Now, ASL is not perfect. Like I said, it's a near ideal, it's not perfect. It's really prone to motion, like I mentioned, but it does have a lot of physiological and also just exogenous agents that can make it challenging to measure it robustly and sensitively in humans. Alcohol, caffeine, just regular changes in blood flow, you know, during the day, so from day to night even. All of this stuff are challenges that can sort of reduce the sensitivity and cause more variability with ASL. And, you know, the community is aware of these challenges. You know, we tend to try to minimize this as much as we can when we do studies. So we try to avoid caffeine. You know, we try to have people stay awake during the scan and stuff like that. So these are just, you know, something I like to put up there just so that people are familiar with when they're trying to do an ASL experiment or try to do an ASL measurement. And again, this is not, you know, limited to ASL. If you're trying to measure blood flow with PET or with any other tracer, some of these confounders are also going to be something you want to look out for. Now, we're still trying to improve white matter perfusion imaging with ASL. I'm not going to spend too much time with this. ASL has slightly some challenges being able to measure deep brain or white matter signal. This has a lot to do with the transit time, so the time it takes for the blood to transit from the vessel into the tissue. And there's a lot of work right now with so many groups trying to improve ASL measurements in the white matter. Let's skip through this. So for the sake of time, the International Society for Magnetic Resonance in Medicine, ISMRM, have sort of worked with the ASL study group, part of the ISMRM organization. And essentially, this is just a group of people that have come together under this OSIPI, which stands for Open Source Perfusion Imaging initiative. And the idea here is to work towards, again, making ASL more open, so making it more accessible, and so being able to sort of standardize and make sure that we have similar imaging analysis techniques, make sure that we have open source tools that allow people that are sort of novice to ASL to be able to do ASL imaging. So, you know, if you are wanting to sort of dabble into ASL, we're putting together an ASL challenge. Essentially, we're going to provide some open source data that people can sort of use to do ASL analysis. There are different task forces. I'm on task force 6.1, but there are different other groups that are working towards making open source tools available. So you can use one of those open source tools and the data that we have here to sort of get your hands wet on doing ASL analysis. A lot of these tools are straightforward to use. I mentioned the Quantifies and Explore ASL. So if you're trying to get your hand, sort of get introduced to ASL, welcome to just look us up, osipi.org, or you can always email me and I'll sort of put you in touch with the groups that are doing the software tool development, or the groups that are putting together an inventory of software tools. So you can use one of those tools to try out the challenge. The challenge should be launched sometime at the end of February, if everything goes well. So again, if you're interested, you can send me an email, or you can keep an eye out on the osipi.org website. And sorry, so I'm just going to end here. One thing I didn't mention is the fact, and the reason why I'm very passionate about ASL is its accessibility. When I talked about PET as being the gold standard, I mentioned the infrastructure that you need for PET. So you need cyclotron, you need the PET scanner, but you also need the people, the resources to be able to run the cyclotron, produce a tracer, make sure the tracer has met quality control, and be able to bring it upstairs and have it, not upstairs, bring it to the scanner and have it injected into the person. This requires a lot of resources when MR does not. Now, in terms of availability, there is one PET scanner outside of North Africa and South Africa. And that one PET scanner is in Kenya, and that was commissioned back in 2019. So there goes accessibility for PET. But when you compare MRI scanners in Africa, there's quite now a number of clinical strength MRI scanners that are capable of doing ASL in the region. And so the idea is to get people in the region that are trying to do some neuroscience work or some neurological clinical studies to start, being able to get up on the scanners and start using ASL. Now, this will require a little bit of optimizing, but I'm more than happy to help anybody out there that wants to start, using ASL on their clinical scanners in the region. I'm willing to help them optimize it and sort of get it going. So just to summarize, ASL, I believe it's one of the suitable alternatives to invasive measurements of blood flow, and it's multiparametric. I quickly touched on the fact that we can use ASL to measure vascular tone, so looking at vasculature, as well as looking at vascular integrity, so blood-brain barrier dysfunction. ASL is quite comparable to the gold standard, both gold standard for blood flow imaging, so O15 water PET or radioactive water PET, as well as the gold standard for looking at brain health, which in this case is looking at glucose metabolism with FDG, which we've sort of demonstrated that. And like I said, there is a concerted effort towards bringing ASL to the clinic. That includes being able to provide standardized protocols and recommendations, open source image processing packages, guidance on how to read and interpret blood flow maps produced by ASL. I think ASL has reached to the point where it is ready to be used in clinic. And there is, I guess the killer applications, if you want to call it that, are looking at cognitive impairment in dementia, stroke assessment, looking at, neuro-oncology with glioma, and actually in epilepsy imaging. And epilepsy is very interesting because in epilepsy, there are some patients that don't actually respond well to drugs and do require surgery. And in those patients, it's really ideal to find where that seizure focus is. So, we can accurately remove the lesion that's causing the seizure and sort of minimize the seizure frequency. The problem is that in some cases, the brain appears normal. So when we tend to do the workup for epilepsy surgical planning, we tend to acquire anatomical MRI images. And in some cases, in about 30% of the time, the brain appears normal. And then what we do technically in the West is we use PET FDG, so we use glucose metabolism, sort of look at physiological changes that are happening in the brain that otherwise appears normal. And this is going to be a limitation for places in Africa where there is no PET scanner. And so ASL provides a good alternative to start looking at doing epilepsy surgical planning in places like Africa. And I'm going to end here, and I thank you so much for your time. Again, this is a high-level talk. I just wanted to introduce ASL to the Udunna Society, and I'm happy to take any detailed questions.
Well, thank you so much, Dr. Anazodo. That was really, really great. I just want to encourage everybody who's joining us live, please to post your questions in the Q&A portion. I see them coming up also. I just thought, I'd start with one of mine. I know you talked and motivated a lot this point of accessibility, and I really liked the plug you put in for even for the African community, how generalizable it could be. And you touched on kind of the computational and statistical analytical kind of infrastructure that is starting to get offline. I wanted to ask you whether or not there's like an additional module that needs to get added to just the standard MRI to have this magnetization pulse added, for example.
No, that's the beauty about, doing stuff like pseudo-continuous ASL. The scanners don't need any additional infrastructure. So, most scanners now come with a head coil, which is what you need to do MRI imaging in the brain. And so the head coil is just standard. It comes with a scanner, and most standard head coils are able to do, to a certain extent, fast-ish enough ASL imaging. Now, most scanners in Africa are typically 1.5 Tesla. So what I'll call the entry-level clinical strength scanners that we have here in the West. A lot of the research that we've done in ASL in the past 10 years have been, quite a lot of them have been on 3 Tesla scanners because it's easier to acquire the images, it's faster. This is where you can tend to play a lot with your parameters. And so a lot of the, if you were to read ASL papers, you find a lot of them is actually done on 3 Tesla scanners. But the scanners that we have in Africa are, as long as they are at least 1.5 Tesla, they are ready. It's just more or less getting somebody on the ground to be able to optimize the parameters. The open source tools that I talked about, and there's quite a lot of them. I just mentioned 2 of the bat. Those are the 2 that I've actually sort of played with. I write my own analysis codes, but those 2 are really easy to use. And it doesn't matter what scanner that you have, they will accept the data and just, they have really good guides on how to work through your workflow analysis.
Yeah, again, this sounds like a really fascinating and applicable tool. So I'll just read this question off. It says here, compared with an fMRI, even in the resting state, how does the ALS MRI solve the reproducibility problem? I think you touched on this during your talk.
Sorry, can you read that question again? I heard gold fMRI and I heard reproducibility, and I want to make sure I'm tying the 2 questions together.
Yeah, I think they're asking with just kind of canonical fMRI, how does this new method relate in terms of reproducibility?
Oh, how well is it reproducible compared to, say, fMRI?
Right.
Yeah, well, it is it's quite reproducible. There's a lot of studies done to demonstrate that. I don't have any of the slides up here. I should have had a couple of them, but I'm going to point out this study here. If you just give me 1 second, I'm cycling through my extra slides here. So this is a study done by the group at MGH, again, demonstrating how ASL provides very focal signal change in response to a finger tap in a motor task. And you can see that ASL is very focal, FDG PET being the gold standard because this is in response to glucose metabolism change. It's quite focal to the primary motor cortex, while fMRI, because fMRI has a lot of confounders, it's not as focal as, say, PET or FDG. And in terms of reproducibility, there are a lot of studies that have compared ASL to fMRI, and they show that they're pretty much quite, the variance is quite similar.
Okay. No, this is great. So I also had another question. I think you really convincingly talked about some of the challenges the technique itself faces about long-term stability and kind of variability and, designing methods for standardization. I'm curious how, you deal with it because it's fundamentally a subtraction method, how from one person to another, whether or not there's a kind of a fingerprint of CBF for me, for you, and for any given subject, how you guys deal with this challenge.
Right, so ASL is a subtraction technique. So you're working with 1% of your signal. And sorry, before I go ahead, I want to correct something. In terms of reproducibility between fMRI and ASL, it's actually, the variance in ASL is actually a little bit smaller than in fMRI, and that's because fMRI has its own added confounders, and ASL is a direct measure. It's the SNR that it's what makes people sort of want to use fMRI over ASL, fMRI having better SNR than ASL. So coming back to your second question about the, because the subtraction technique and some of the challenges that I talked about, the signal that we're working with sometimes is an order of 1% signal difference between the label and sort of the control experiment. So what we tend to do is to do repeated measures. So we repeat that control label paired, experiment multiple times. And by repeating the multiple times, we're able to sort of minimize inter-person or inter-subject variance. And also it actually allows us to sort of boost our signal, for lack of a better word. So you have to do that control level experiment multiple times, at least a minimum of what's equivalent to 5 minutes on a 1.5D MRI scanner.
Gotcha.
Now there are fast, so people are not coming up with fast approaches to sort of condense that time using deep learning. Deep learning to me is, I'm still trying to figure out what it is. It sounds like voodoo, It works. We don't know how it works, it's something, so, but there are really good approaches that people are trying now to do with deep learning to minimize some of the confounders, motion being one of them, because it's very sensitive to motion. So if there's any difference between head motion, or even physiological motion, breathing patterns and stuff, between one paired experiment and the next, so one repeat and the next, it can sort of affect your signal or your SNR, your signal to noise ratio. So people are working on ways to sort of compensate for that.
Yeah. No. These are. I know, as you know, when a new method comes up, these are kind of fundamental questions that end up needing to get resolved. I just want to continue to encourage everybody to please post your questions in the Q&A. I just want to follow up this kind of SNR versus temporal resolution question that you had. I'm curious if the field is also considering kind of increasing local kind of resolution by doing the magnetization at a common artery to just a very specific local area. I don't know if you guys are considering.
Yeah, so that's how it started off. It started off with continuous Atherospin labeling. That's essentially what you did. You had to sort of use a coil to transmit your signal, and then use a separate coil to acquire, read out your signal. So Castle, that's how we started. But it's very it's time consuming to do that type of experiment. Typically don't get much with Castle compared to what tends to be now the common approach, which is pseudo-continuous, which is a variation of the Castle technique, except you don't need that two-coil system. You can sort of do continuous pulsing of your area of interest. In this case, it's in the neck. Mind you, ASL can be done anywhere in the body. People do it in the kidneys. It's very straight. The vessels are straight up, so it's easy to do. So it started in the brain. It's very common and sort of well established now in the brain. But we started off with doing that too.
Okay, this is great. I just want to say that the reason I asked this is because, there's a lot of people, because of the clinical relevance, there's deep brain CBF is not necessarily studied as extensively, and a lot of people focus on the cortical mantle. And I'm curious how you can resolve this SNR problem in these smaller structures by doing really focal measurements,
Yeah, so you don't really need focal measurements for that because you have to remember that the feeding vessels go up into the brain and just, go through the vascular territories. And so one issue with doing ASL, if you're going to do one labeled measurement where you use one bolus to read out your whole brain signal, is you might end up having the problem you're describing, where you get into the deep brain and you might have transit time issues. And one way of getting around that is to do what people sometimes do, multiple post-labeling delays, where you read out your signal as it travels into the brain, or multiple I'll keep it simple, to multiple post-labeling delays. And that's one way of being able to interrogate your signal as it decays into the brain. And so you can read out vascular differences and be able to measure the deep brain, for example, better. There's also velocity encoded ASL, there's velocity dependent, and also allows us to measure some of those deep brain changes better. So there are techniques around being able to sort of work around this problem.
Awesome. Okay, so we don't have. So I have actually.
I could take two more questions. I could take two more questions.
Okay, sure. So maybe I have another one of my own. This is a little bit higher level. I was really intrigued by this thing you mentioned earlier on. For example, in your postdoctoral schizophrenia study showing, and you also showed some slides that show their CBF deficits before even some cognitive or behavioral deficits. I'm curious if there are hypotheses that suggest that these CBF deficits themselves also kind of cause or exacerbate the pathology itself, which people think are coming from the neuron side, basically.
Yeah, so that's a vascular hypothesis in Alzheimer's disease. So almost almost every disorder now is trying to look at this vascular hypothesis. So there's sometimes uncoupling of blood flow so that when there's not enough blood flow to meet demand. There's so many hypotheses behind that. Some of it has to do with glutamate excitotoxicity. When you have an upregulation in glutamate, it's driving up energy, not enough to meet demand. Inflammation being one of them. So astrogliosis or microglia activation being one of them that drives up energy and not enough to meet demand. And so I'm interested in trying to understand, at least in the dementia context, inflammation and vascular dysfunction, and more or less looking at breakdown in blood-brain barrier, which is something that the inflammation, chronic, low-grade chronic inflammation can do. And that may also lead to this vascular dysregulation or uncoupling of blood flow to meet neuronal demand.
Yeah, no, this is really, really great. Thank you so much again on behalf of SONA. I just want to say thank you very much. I want to encourage the community that are going to watch this on YouTube and various other post-recording. Please reach out to Dr. Anazodo to get this going in terms of how it applies to your own research. So once more, thank you very much.
Thank you so much.
All right. Bye-bye.
Bye.